The title of this presentation is From Computational Biology to Data Science into Engineering. To begin with a presentation overview, we'll be introducing interactions between computational biology and data science as a starting point, including just some overview of a couple of algorithms that have been discussed on this channel. But rather than only reiterating couple of the several characteristics of these algorithms and what their predictive capabilities are and how that relates to other types of generative AI or other types of AI workflows that have really been gaining a lot of prominence recently in industry and in academic research. We're going to also be able to describe these types of algorithms from a different perspective and being able to form several open directions of interest for current research, either within industry or academia, which relate to interpreting biological data and trying to obtain some type of meaningful inference and meaningful output from running different types of algorithms on sequencing data or other higher dimensional complicated data. And so, as we're saying in the second bullet point of the overview slide, from the wide variety of algorithms that we describe for the purposes of computational biology, we're going to draw the attention of the viewer to broader themes in data science and engineering, and how this relates to data engineering and making use of some next generation higher throughput data, like how we've been seeing recently with the advent of newer sequencing methodologies and newer sequencing technologies and other types of data acquisition methods and assays that can be used to describe various biological phenomena. So I first wanted to begin with transcriptomics related algorithms because these algorithms are very interesting because they very much make heavy use of probabilistic computation, and they're looking to exploit certain sequencing data, whether it's something that's gathered from some type of experiment gathered in the lab or other types of biological characteristics that can be inferred or gleaned from some assay or some type of continuous development of assays from experiments. And Trinity is helpful because it's one such open source workflow that I've used and described in other types of videos on the channel, which can also be interfaced with Callisto, which is an open source software package from a lab at Caltech, which is able to make use of a primary step of pseudo alignment. And the pseudo alignment step allows us to determine differences and similarities between various sequences that are generated from a transcriptome or genome assembly, which can then help us determine whether, look at the output from other statistical measures of comparison or other statistical metrics, which can help us characterize either genetic similarity or dissimilarity, as well as any potential other assays or experiments that could be used to test other characteristics that maybe couldn't be gleaned just from the sequencing information. And for Trinity, for the transcriptome assembly workflow, we very much need to think about which types of input files do we need for predicting on the order of millions of genetic targets, as well as what are the type of output files that are produced. And this helps us think about how do different genetic targets reflect the bulk or single cell RNA sequencing and the resolution that is gathered from experiments. And this kind of difference between single cell information and bulk information, whether it be like for RNA or many other types of systems. I'm going to bring it up and reiterate it more near the end of the presentation with a list of open questions which are relating to the themes and how they relate to data science, because it's very much like reflecting some type of fundamental difficulty in that if we have some narrow type of interpretation of some biological information, when we're feeding this into either machine learning, deep learning, or other types of algorithms, the output that we're going to be able to get from these types of from this type of training set data is very much going to maybe reflect a very restrictive notion of whatever phenomena we're trying to study, because it's just gathered in one instance or across one line of cells or across one group of cells, which maybe can exhibit some types of characteristics which are important for the progression of a disease or other types of circumstances like genetic editing and so on. And as we mentioned in the last bullet point, we're going to make use of Callisto, which is another workflow which can be used to quantify the abundance of transcripts that's produced from the transcriptome assembly pipeline, because it's qualifying as one downstream process that we can perform with the fast output, which is just a list of potentially hundreds or even in some cases from execute from simulations of Trinity that I've ran, you can have hundreds of thousands of genes output in the Trinity transcriptome assembly. And it's very much helpful to look at different types of ways for either maybe visualizing how genes are organized and how this can reflect upon characteristics in mediums or modes of interaction within the genome. But for Callisto, which is the secondary downstream process that we can perform from Trinity output, we're interested in asking about which processes are performed for generating the IDX file from the transcriptome. So if we feed the transcriptome FASTA assembly as out as that which is generated from the Trinity output to Callisto as input, we need to think about how we can generate this type of intermediate type of file on which the algorithms for Callisto will perform, which are primarily relying on pseudo alignment, will be performed. And this is related to the fact that we can also think about at which steps of the Callisto workflow are the pseudo alignment performed. And this is also demonstrating what is the primary metric of output of Callisto, which is, as we're going to discuss further, it's relating to the probability or of obtaining a certain transcript within all of the set of transcripts or the transcript abundance. And other bioinformatic pipelines, including Augustus and like genome and like other algorithmic pipelines for or workflows for gene prediction relate to predicting the sequences of the transcriptome and genome, because we have several types of processing steps for Augustus too, which very much make it like a very simple use case for data science, because we're making use of some type of machine learning workflow that's given off the bench. And from just making use of the various other files that the developers from Augustus provide, we're able to make use of a hidden Markov model, which in itself is a type of probabilistic program, because it's looking at the probability of obtaining several different connections or mediums of or modes of interaction between different nodes within a complicated network, which can be scaled up to networks with potentially thousands of nodes. And this is relating to some type of machine learning workflow, because we can look and add on further other types of features to machine learning architectures, like how we'll describe and summarize in the slide at the end of the presentation with a series of like several open questions and directions of research interest, because we're looking at how the classification from the machine learning output is related on just some open source architecture in which someone built some type of network that could probably be mapped onto some neural network with some fixed number of neurons and which has some associated activation threshold depending upon how the hyperparameters are initialized at the beginning of the classification or training, depending upon whether the neural network or other type of model is supervised or not. And we can also look to make use of some open source platform from the NIH, which provides a basic local alignment search tool, which allows us to be able to search through our potentially exponentially large high throughput gene space, which can help us determine whether there are any types of common conserved sequences, or we can also make use of some types of characteristics of the sequence for variant detection. And the Trinity workflow, which is getting also to connections to several connections with the genome workflow that was given through Augustus, it's a little bit of a step up in complication from the workflow that was given from Augustus on the previous slide, because you have to make more use of data preprocessing steps for looking at either RNA or DNA sequencing files, which are going to be fed in from a sequencing source, which is given by Inchworm, which is given by the initial stage of the algorithm. After which, in the second step in Chrysalis, there's a lot of additional manipulations performed from k-mers, because k-mers are relating to initially isolated segments of the genome, which are going to be overlapping potentially over on their edges. And from these overhangs or the regions of the genome over which these different reads overlap, Trinity, as a transcriptome assembly algorithm, it's making use of Chrysalis to form contigs and being able to denote or assign different contigs within clusters, which are then used to construct de Bruijn graphs. And there can be a lot of different types of ways that you can look at de Bruijn graphs, or being able to visualize different steps of the intermediate output from Trinity, because when you're running the transcriptome assembly or any other type of more complicated algorithm, it generates several type of intermediate files, whether it be from the beginning of the algorithm, which is indicated with the sequencing source, from this RNA extraction step, which is one potential source of sequencing that we could have. It performs several checks on the formatting and the arrangement of the files within the FASTA input, so that it makes sure that as it's performing any other potential down processing steps for the transcriptome assembly from either chrysalis or butterfly, which we're going to describe in the third major step, it's making sure that it can form the contig clusters appropriately, as well as being able to generate some notion of the probability of that the algorithm is certain that some type of sequence that it outputs in the FASTA file that's produced in the culmination at the end of the transcriptome assembly is in fact with very good chance the actual sequence that could be experimentally investigated further and realized in the lab. So it's very much allowing, experimentalists to accelerate their workflows, and it's relating to data science within several types of industries, because it's either looking at trying to maybe synthesize some types of compounds of interest which perform a desired function more rapidly, or there could also be some other type of potential application in looking at how to perform a directed evolution of some enzyme, which is relating to the previous application. But as a third potential application and something that's a little bit more distant from the two, you could also look at either looking at, running some type of molecular docking workflow, like some other classes of algorithms I'll describe later in the presentation, which will relate to several types of open questions and questions of research interest, which can inform assay development or or even three-dimensional organization of the genome. And so given in the third step of the Trinity workflow for transcriptome assembly, in the butterfly stage, the algorithm is finishing its compilation because it's performing a transcriptome assembly into a multi-FASTA file. And by performing a T-BLAST, which is another variant of the basic local alignment search tool that was given from the open source NIH platform that I described in the previous slide, and as we were motivating different types of genome prediction algorithms, we're able to isolate potential gene candidates from generated transcripts, which can, as we had alluded to earlier, it can inform assay development for further experimental investigation. And in the fourth step, besides the first three steps of the Trinity workflow that we've been describing, as we had mentioned, we can look at various ways of visualizing de novo assembly graphs, which is imparting upon us some type of probabilistic knowledge from our computational biology or transcriptome assembly or bioinformatics pipelines, because it's allowing us to determine from a FASTA file, which we feed as input, what would be the certainty of the algorithm that it can make an appropriate or well-guided classification based on some training set data or initial input from the RNA extraction or other sequencing source. And the parameters that are fed into Trinity before processing in the first introint stage, as we had alluded to in the previous slide, it relates to the facts that, as given in the left here, we have to make some bioinformatics pipeline call with Trinity, as well as in the fact that we have to specify the input format of the sequencing, whether it's in FASTA or FASTQ, which are very types of related inputs, but they have some slight differences and intricacies in how the sequencing information is stored in the FASTA versus FASTQ format, which I won't get into here. And then we also have to specify two additional parameters, which are relating to the set of the left reads and the set of the right reads, as well as the total number of CPUs that we're going to allot for any one of the computations which can be performed from the transcriptome assembly from Trinity, which sometimes, as I've done in some simulations of Trinity that I've ran, it can take on the order of 4 or even 5 days. So this is why it's important to have in mind the max memory parameter, which is given as the last argument, which is fed into the Trinity bioinformatics pipeline call, because it's relating to how much memory Trinity can make use of when it's performing various steps, as we had exhibited in the second stage in chrysalis, when it's looking at k-mers and performing manipulation of k-mers, so that it can determine what are the regions of overlapping reads from which it can form some more global picture of the transcriptome from these smaller reads along isolated segments. And from the steps 1 through 3 of the workflow that we had described, we can provide some broad summary of the Trinity output, which is relating to several types of uses within data science and being able to look at either how to perform a more quicker and faster search and more efficient search of a larger data spaces, which is given by the sequences which are generated in the transcriptome. And then this is also relating to other aspects of computational biology and bioinformatics, because we're making use of several qualities of either an RNA sequencing data source or a DNA sequencing data source, which can each be characterized depending upon how many unique sequences which are detected by inchworm in the first stage of the transcriptome assembly algorithm or pipeline, because it generates it reads in initially approximately 400, a little more than 440 million unique sequences. And after processing all these sequences, when it passes through Casalis, it performs the clustering stage in which it clusters millions of sequences together from the previous stage into disjoint transcripts. So this is very much related to the type of graphical structure, which is relating to the type of probabilistic knowledge that I had alluded to that we can gather from a transcriptome assembly pipeline in the previous slide. Because even though it may not initially appear that there's some type of interpretation in terms of a probabilistic program or probabilistic computation or probabilistic certainty or inference, it's in fact relating to all of these types of notions and interpretations of probability, because we're able to generate some type of graphical structure, and it's helping us determine from a data science perspective several different types of structures that we can study the characteristics of, such as what is the node amongst each one of the base pairs of a sequence that's generated in the transcriptome assembly, and what's the degree of this node, because if a node has a certain higher degree within a sequence, it will indicate to us that perhaps the Trinity transcriptome assembly, given some set of input sequences from the RNA sequencing or other molecular RNA sources, can encounter some difficulty and potential bottlenecks in the algorithm that would hinder its classification ability. And this is very much relating to, as we have been alluding at several different types of presentations already for some set of open questions, how this relates to different types of notions of interpretability in machine learning, and how that can relate to the types of data sets that we're feeding into these algorithms if we're looking to simulate or compile simulated or supervised workflows. And from the butterfly step, which as we had mentioned in the previous slide for the workflow, is given by the final assembly of all the transcripts, the mean gene length across all of the that is the total length of all of the base pairs that are assembled in the transcriptome divided by the number of genes was approximately 376 nucleotides long out of approximately 134,000 transcripts that were produced. And for another RNA sequencing data set too, we can see that the numbers that are generated as performance metrics along each one of the 3 steps that are given from the trained transcriptome assembly can differ, in which in this case we had approximately 371 million unique sequences, but then in the Christella stage, the graph structure had, give or take, 100,000 less vertices and almost 100,000 less edges due to the fact that there was less complexity in the mRNA extraction in the RNA sequencing data set that we fed to the algorithm in this instance. And then from the butterfly stage, which again is relating to the final assembly of all the transcripts into a multifaceted file, we were able to conclude that overall, even though the total number of genes that was produced in this compilation of Trinity was a little bit less, the average length or the mean gene length was a little bit higher, from around 109,000 transcripts that were produced. And I thought that was very helpful to describe connections between data science and computational biology, and how it can allow more theoretical people and experimentalists to interface with one another. Because the steps that I described previously from the transcriptome assembly can be repeated for any source of RNA sequencing for generating hundreds of thousands of new sequences, which is to say that maybe depending upon the types of organism or host genome that you're looking at, you may be uncertain about what could be some closely related family of sequences which are relating to what's gathered in experiments, which could either inform your assay development or further accelerate your experimental efforts, because you could be able to generate some types of algorithms from these probabilistic routines that we've been describing, whether, as we had mentioned, it's being related to probabilistic inference, um, you know, um, the certainty with which the transcriptome assembly algorithm is able to generate sequences within the multi-FASTA file that's assembled in the butterfly stage of the algorithm. And this is also to say that the FASTA output has many applications that we have been alluding to in genomics and transcriptomics and related algorithms. And there can even be other classes of algorithms which may not necessarily relate to classical computation alone, like I'm going to mention and allude to further at the end of the presentation, just to describe maybe other potential directions of future research in which many people hope within industry that these types of algorithms could lead up to some speed up in runtime without accumulating some type of loss in classification ability of these like machine learning or deep learning workflows. And I wanted to also draw for their attention to another open source pipeline or a short series of scripts that are open source, which is providing a way for naive diversion graph to be constructed in Python, which is relating to another presentation that I gave on the channel, in which I describe very specific characteristics of the output of these types of algorithms, which is relating to the second stage of the Trinity transcriptome assembly pipeline, which is relating to clustering contigs together so that some type of diversion graph can be formed from which separate transcripts can be produced in the multi-FASTA file assembly. And this is, again, very much relating even to a different type of aspect of data science, because a lot of data scientists or applications within classification that make heavy use of algorithms that data scientists use, which essentially, all relate to some type of, regression or basic statistics. It's relating to the fact that we can look at how different types of biologically oriented information from sequencing resources or FASTA files can be used to generate some type of helpful or informative information about the behavior of the biological system when it's under other different types of stress and strain, which can potentially impair its ability to carry out its native function. And the output from the GitHub script that was given in the repository was that if you're looking to make use of their script, you have to feed it some type of FASTQ input, which, like we have mentioned, it's imperative because when we're constructing a diversion graph, we're looking at different sequences that are produced from either a transcriptome assembly or a genome assembly pipeline, and to have some type of handle and some type of assurance on the statistical precision and the statistical certainty that we want to use when we're looking at different types of these FASTA sequences within potentially hundreds of thousands of sequences within the FASTA file at the time. We have to look at the common elements which belong to each one of these sequences in the FASTA file, and then how that can be fed in as an initial input file to the parameter or to the open source workflow that I demonstrated from the two developers on the previous slide for parsing together some type of local information from the shorter length reads and being able to stitch this up and build this up into a larger global information about the genome or the transcriptome. And there are countably many sequences from the GFA file that are produced, in which from this FASTA file that's given as input, we can say that we can print out the contents of the GFA file, which, as we had mentioned, it's given by a file format that's produced as intermediate outputs in several types of bioinformatics and computational biology algorithms, because it's relating to the fact that it's carrying in repackaging the information that's given in the FASTQ format that we fed in as an initial stage for the naive diversion graph construction, which then allows us to proceed further to obtaining our diversion graphs, because it's packaging the sequencing information depending upon the overlaps between the reads, which is going to inform the number of sequences which are which are contained within each cluster. And this is ultimately also influencing the total number of contigs, which would have to be processed as was given, as it was described in the second step of the Trinity transcriptome assembly that's going to be used to construct Debrugian graphs. And the contents from the GFA file are used to construct a FASTA file, in that the FASTA file, unlike many types of tutorials which describe how to perform data wrangling or data manipulation from FASTA files, don't really describe a lot of the specific contents which can be given from FASTA files or FASTQ files that are fed from into bioinformatic pipelines. So on this slide, I thought that was really helpful to actually demonstrate how the FASTA file is structured after it's passed to the naive Debrugian graph construction, and how this is relating to the contents of the GFA file, because the GFA file that we obtained from the FASTA file, its contents are given by the fact that there's some type of unique identifier which appears before each one of the sequences. And you can see that different types of information give various indices pertaining to the length of different types of ends of the sequence, as well as the length of the total number of length of base pairs that's given in the sequence, which is important, again, as we've mentioned, from the output that's given in the Trinity transcriptome assembly, several types of performance metrics relating to how many k-mers are there in the chrysalis stage, what is the total number of unique sequences, or what's the mean gene length, or whether there are any contigs in which perhaps the average gene length within one contig in comparison to all of the other genes which are given in another contig could be higher, which could exhibit some type of performance bottleneck or increase in computational complexity of the transcriptome assembly or other types of assembly algorithms when it's being simulated and compiled over a certain part of the RNA sequencing or mRNA extraction data set that's fed into the algorithm. But furthermore, the contents of the FASTA file parallel, as we have been alluding to and describing within several different approaches and themes of data science, they parallel the contents that are produced from many transcriptomics algorithms. But it's very much helpful because now we can have different types of contents and characteristics of the file, which can facilitate data visualization, such as I've described in another video on this channel, which is relating to making use of a potential downstream algorithm for transcriptome assembly, which is given by Bandage, which allows us to look at different types of clusters and how they're represented in the sequence space from the total list of sequences that are given in the multi-FASTA output that's produced by the transcriptome assembly Trinity. And now I wanted to switch a little bit of gears towards another type of bioinformatics related algorithm, and how still, even though obviously it's not very much relating to the transcriptome assembly or the data visualization or the genome assembly that we had described earlier, how very much it still relates to some other potential bottlenecks that we're going to describe further through the list of open questions at the end of the presentation, and how classifier design and classification and the certainty in which we perform classification, those themes of data science very much in, how that relates to biological systems still very much appears in a similar type of format, and maybe just under a little bit of a different guise. Because this is relating to orienting ourselves with Autodoc Vina, which is, as we had alluded to earlier in the presentation, it's a molecular docking workflow, which is relating to PyMol or other types of Autodoc tools that are publicly available, that allows us to set the atomic coordinates about some region of interest from which we can perform a molecular docking in silico experiment that can allow us to determine what are the types of characteristics of electronic structure or other characteristics relating to the structure of the receptor that was given for some disease of interest, which can help us obtain some other type of biologically oriented information relating to data science. And as mentioned in another presentation on the channel, to make use of this open source molecular docking workflow, you have to follow pre-processing steps for the in silico approach, which will allow us to convert PDB to PDB-QT files within PMV, within the Python molecule viewer. So this is very much to say that when we perform some type of classification task, a lot of times, even like we saw with the input that was given for the files in the transcriptome assembly, there can exist several different types of pre-processing steps that we have. In this case, the pre-processing steps for molecular docking or in silico workflow, it's more relating to the fact that we have to make sure that the atomic coordinates and other in other formats within or other features within the file, which are indicating where bonds between within atoms of a chemical structure connect together, have to be specified in a very precise way, as well as other pre-processing steps that I've described more in this presentation on the channel. But returning to our primary topic of discussion with the transcriptome assembly workflows and algorithms, the workflow that we are talking about, which is describing several types of structures of graph and visualization, which is very much relating to visualization that we have to keep in mind when looking at different properties and characteristics of electronic structure. It's very much manifest because, as we had mentioned, from some type of intermediate output that's given from the k-mers of overlapping sequences, we can discuss in this slide from the perspective, as we've been mentioning, of data science in statistical inference and statistical certainty within computational biology algorithms, this performance benchmark, which is very much relating to the total number of contigs, as we had alluded to, and the total number of k-mers, which are given in the Dobrugin graph. And this graphical structure is very much helpful for data scientists in the field of computational biology, because, as we had mentioned, you can get an idea about what is the certainty or probability of a classification being made appropriately from some certain sequencing information that a sequence either could maybe have some type of conserved sequence or other characteristics, depending upon the total number of contigs which appear in the Dobrugin graph, as well as the total number of k-mers which appear in the Dobrugin graph, which is ultimately relating to additional pre-processing steps that occur earlier in the transcriptome assembly workflow, which are pertaining to the total number of unitic split, which is ultimately relating to the unique number of k-mers that are first read into the algorithm. So we can see that because we have the FASTA file and the FASTQ file, which we had mentioned is a file which appears in several types of data science situations and data science applications and bioinformatic applications, because we're able to read in this total number of unique k-mers, which is then assembled, or it's assembled from potentially a transcriptome or a genome assembly pipeline, like in that FASTA, in the contents of that FASTA file and the GFA file that we had described when we were making use of the open source implementation for the naive Dobrugin graph, and how this relates to the complexity of the sequence space and the space of sequences within the transcriptome or the genome, in which we're trying to establish some well-guided inference, because we can have easily on the order of several million k-mers which appear in the Dobrugin graph, which already makes this a very good type of non-trivial and complicated higher dimensional data structure for a data scientist to work with. And the Callisto workflow, as we had mentioned, which we can further describe, which is going to relate to some other potential data structures which can be of application and of use for data science, is that we have to make use of some pre-processing steps to build some sequence of .idx files, which is relating to building Callisto indices. And as we had mentioned, with the fact that, or with the observation in mind that the primary processing step of Callisto is relating to the step of pseudo alignment, we can perform additional steps by letting the algorithm run on the IDX files as input, which will allow us to perform a quantification of abundance stage, which is given by the same type of quantification stage or quantification call to Callisto that I made, which is given in the open source documentation from the Pasher lab that they provide for Callisto, in which they describe the input, which, as we had just alluded to, it's given by the IDX file, which is some transformation and pre-processing of the FASTA file from either the transcriptome or genome assembly, as well as the fact that the output file is given by some fasta.fastq.gz file and another reads file, which can allow us to have some type of information about the quantification of the transcripts from the information and the transformation of the contents of the FASTA file, which are given in the IDX file. And from the quantification of abundance, we can have some type of target ID, which, as we had mentioned in some of the performance metrics that are given for Trinity, we can look at several types of complexity measures which are given for the output of this algorithm, whether it be from the length or the effective length, as well as the estimated counts or the TPM index, which stands for the transcripts per million. And then as from some open source output that's given from Callisto that I placed on my GitHub several months ago, we can have a bunch of .tsv files which are representative of the output which is given from the Callisto pseudo alignment steps and from the IDX files which are generated as the intermediate output. And this is given by the fact that from the Trinity transcriptome assembly, just out of curiosity to see how certain types of parameters vary that the developers in the Pasture lab described for the Callisto workflow. I looked at the transcripts per million, the estimated counts, as well as the length and the effective length for some fixed number of target IDs that were given from genes that were initially assembled in the FASTA input that was fed into Callisto from the Trinity transcriptome assembly. So this is giving another type of data structure and another type of biologically relevant fact and criteria that can be related to and very much, related to experimental efforts and very much of interest for computational biologists, because you can look at what is the range of the length across some subset of genes, what is the probability maybe of interaction between some type of genes which could share in similar characteristics, which besides conserved motifs can also be related to the fact that maybe one gene can be really long or another gene could be much shorter, which could generate which could indicate to us perhaps some type of feedback interaction between more complicated genetic networks, which is dependent upon the estimated number of counts, as well as the probability that we would find this transcript in a collection of 1 million of transcripts, in a collection of 1 million transcripts. And from this quantification on abundance stage that we have been describing in the various performance metrics that are associated with the quantification of abundance, we can perform in the third step additional downstream processing, which is related to the fact that for the basic local alignment search tool, we can isolate, as we have mentioned in the case of Trinity, isolate potential gene candidates for generated transcripts, which can inform assay development for further experimental investigation. And the quantification abundance can also finally be summarized with the final table, which is given for another performance benchmark for the Callisto quantification, in which we can see that from the total number of reads, there's a type of monotonic increase in the complexity of the runtime and the complexity of the total number of reads, which have to be pseudo-aligned from this Trinity, from the Callisto algorithm that we have been mentioning, as well as the fact that there has to be some finite number of iterations or rounds of an expectation maximization algorithm, which is allowing us to make some type of conclusion about the quantification of the abundance of transcripts from the initial FASTA file that's given as input into Callisto from the total number of reads were pseudo-aligned. And we can see that just as some very as some big advantage of Callisto, it's able to run extremely quickly, even from the number of reads being more than 150,000. So it can potentially easily handle a pseudo alignment for on the order of millions of genes, which could then be used to gain some type of insight into more complicated genetic networks. But besides Trinity and Callisto, with open source molecular docking workflows, we can characterize structural aspects of compounds of interest. And this was given by the fact that, as we had alluded to and described earlier with AutoDocVina, we can make use of similar type of either sequencing information to construct chemical structures and to study aspects of the electronic structure when different ligands and receptors interact within some specified range of molecular and atomic coordinates. And this is just given by the fact that out on this slide here, we have an example of several types of conformations which are produced from the open source implementation of AutoDocVina that I ran, and then by using various other open source platforms for visualizing FASTA and FASTQ output from these algorithms, we can look at the spatial arrangement of these compounds within three-dimensional space, which would have some type of biological interpretation for interactions, either like protein-protein interactions or even ligand and reception interactions on a smaller scale. And as we're getting to the last slide, I wanted to provide, as I had alluded to, a sequence of several open problems and questions which are relating to the themes within data science and the computational sciences, like we have been mentioning, and how and trying to see or foresee effective ways for experimentalists and more theoretically minded people and data scientists to interact. And this is to say that moreover, to systematically characterize structures of hundreds of compounds at a time, and also to analyze other type of biological phenomena. New directions of research can be developed from any one of the following directions, in which we have 10 types of items, which we're going to spend a couple of minutes discussing just more near the end of the presentation, which can help tie the themes up of data science that we have mentioned, and also kind of summarize things more densely within one slide, which is given from the first item, which is relating to classifier design, and it's relating to how do we tailor training set data for machine learning or deep learning output to prevent spurious or ill-informed output. And this is probably very much going to still be a type of question of research interest and computational interest for years and years to come, because even if we do get more sophisticated algorithms, if we're always looking at a supervised algorithm, it's always going to be dependent upon some degree of the training set data. And for instance, from the total number of conformations which we have listed, which we have provided images of in the previous slide here, we can see that if we want to run some type of automated version of molecular docking or in silico workflow, that if we don't have some type of information about a new type of structure that's being fed into algorithm, the algorithm could perform very poorly on a classification task on this type of structure, just based upon the fact that we didn't feed anything remotely close to that structure in the training set data. So that's a big problem in machine learning and deep learning workflows, which it's probably always going to be of interest to investigate from different use cases. But in the second item, we can also think about what are the performance bottlenecks for training or simulating machine learning or deep learning frameworks. So the thing with machine learning and deep learning frameworks is that in many cases, these type of frameworks, as we had alluded to in the first questions, a lot of times they can have many very complicated pre-processing steps for either the input files or other types of intermediate output that's generated, which can really hinder many people from being able to just take these types of frameworks and networks off the shelf and being able to use them and apply them in other types of biological contexts that they could probably be used for positively and contribute positively towards in terms of classification accuracy. And three, would quantum extensions of classical algorithms not be able to probe qualities of compounds with quadratic or exponential speed up and runtime? And this is very much relating to the molecular docking workflow, because a lot of people in the field, even relating to quantum computing or quantum chemistry, have really been trying out their algorithms on many different types of compounds. And it's always interesting to see if there would be many, or how broad the space or how wide the space would be in terms of how many sequences would there be of compounds that could be much more quickly analyzed from a quantum algorithm, which could potentially give some quadratic, or in the most optimistic case, an exponential speed up in runtime. Or even for the fourth item that's given in this list of open questions, are there any novel statistical measures on molecular structures that can help discriminate properties of different materials from one another? This is very much related to a lot of topics that people are interested in, with relating to directed evolution of enzymes or directed evolution in general, because there are several different types of materials that can be produced or are byproducts of chemical reactions. And it could very much be helpful to have an idea about what types of statistical properties or characteristics or bottlenecks of these processes, which could lead to producing novel types of materials, or even novel types of green materials at scale. Or for the fifth item, how would one compare the output of different algorithms for molecular enzyme discovery to currently deposited compounds or on AlphaFold or the Cambridge Crystallographic Data Center? And this is very much related to making use and looking at whether there's any information from publicly available depositories like what we had mentioned from like information that we can get from the PDB files and the PDB structure from the protein data bank, and comparing it to either AlphaFold, which is maintained by Google Brain, or either the Cambridge Crystallographic Data Center, which is having an a collection of I think a little more than 1 million small molecules, which is helping us, which is helping inform our molecular enzyme and discovery efforts for discovering properties of new compounds in a high throughput manner. Or even in sticks, we can look at for other classes of bioinformatics centered algorithms rather than those for molecular discovery. Are there any novel probabilistic programs that can reduce the uncertainty of producing output sequences, such as those from the FASTA file of genomics and transcriptomics workflows? So this is very much related to, as we had mentioned, that there's some type of probabilistic uncertainty when we're generating different types of sequences, which are generated in the FASTA outputs from the multi-FASTA file assembly, which is given in the butterfly stage in the last step of the Trinity transcriptome algorithm. And then we're trying to very much see about if we can have some type of better handle or control about the uncertainty in which certain classification or sequences or tasks are performed from machine learning like algorithms. And in 7, we're thinking about how complicated are novel molecular structures and what would be the approximated polynomial runtime of current algorithms for conducting initial in silico studies independent of experiments. This question is pretty much more explanatory because if we can obtain some type of other polynomial runtime algorithms, would there be any type of novel molecular structures that we could study further? And finally, or in getting closer to that, not finally, but in the 8th item, the 8th question, how can language models or generative AI models be leveraged in this biologically focused context, which is very much related to the 7th question? For the 9th question, how can notions of interpretability for large classes of AI models be related to the 8 questions above, in which we can either think about quantum algorithms, machine learning algorithms, deep learning algorithms, molecular docking in silico algorithms, and then trying to maybe compare against some publicly deposited information for compounds like in AlphaFold or the Cambridge Crystallographic Data Center. And then finally, in the 10th question, in the last item of the list, to conclude, can single cell measurements be reliably fed as training set data for determining experimental characteristics of bulk data? This is also relating to some questions that and some topics of our discussion that I phrased earlier in the presentation, in that we were discussing about how even if we're running a supervised algorithm and we feed it training set data, if we feed it that training set data that's primarily just relying on single cell measurement, there can be a drop off in the classification in the reliability of our classification of this model, because it's very much running into types of difficulties because there are several types of single cell measurements that we can gather for several types of cell types, or even cells that can be under certain types of diseases or under the impact of different diseases or infection from a virus or different conditions, which could impact what type of information we would hope to obtain in terms of from a classification from the bulk data that we're trying to study from the single cell measurements that we have. And so the concluding remarks that we have is that we introduce interactions between computational biology and data science and engineering, including transcriptomics, genomics, epigenomics, molecular docking, as well as phylogenetics, in which we can look at the probability of accumulating mutations across different generations of computation and how that informs our classification ability and the statistical certainty in which we can make certain conclusions about our biological systems. And from an overview of the classes of algorithms above that we have been discussing for the presentation, we introduced several areas of difficulty in the data processing, data manipulation steps for computational biology, bioinformatics, genomics, and transcriptomics workflows, which can even have implications for several other types of machine learning, deep learning, and quantum algorithm workflows, like we had alluded to in the previous slide with our set of 10 open questions. And then we ended the presentation with highlighting connections with novel algorithmic development for approaching a wide variety of analysis-related problems. And thanks for watching.